Hello and good evening, everyone. We will be starting shortly. Okay. So is my screen visible? Hello. Is my screen visible? Okay. I will keep on. I will keep on admitting people as they join. So you can ask your friends if they are around to join as soon as possible. And to all everyone else, welcome to the week 2 lecture on animal physiology. This is a PMRF-NPTEL problem solving session in which, as we had discussed in the previous class, we will be solving various problem questions from the previous year's assignment of this course. This week we have the lectures on integumentary system and the skeletal system, and we will be following up on this. Okay, so just a brief introduction. My name is Anubhav Chatterjee. I am a Prime Minister Research Fellow and PhD scholar at the Developmental Neurobiology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. I am the TA for this course, and you can address all your questions to me either directly in chat. You can either post them on the NPTEL forum that is there, or you can go right ahead to my YouTube channel. So as discussed for everyone who was here last class, we will be having live recording of these sessions, and that will be in totality uploaded to this YouTube playlist, which you can go ahead and refer to in later points. You will also be having all the PowerPoint presentations that are being dealt with uploaded to you, and that link is available at your NPTEL course site. Is all of that clear? So if anyone has any questions from the last week's lectures or anything you want to ask from this week's lecture, then we can deal with that now. Otherwise, we will going ahead with this week's problem solving. If anyone has anything to ask. Okay, so I will just briefly summarize this week's lecture that we had already covered. We have been learning about the integumentary system, which is the system which essentially covers the external surface of our body, which essentially includes skin and various accessory organelles, including the hair, the sebaceous glands, and the sweat glands. Uh, Going into a bit detail, we have learned about the various aspects of skin, and that skin has three different layers: the epidermis, the dermis, and the subcutaneous layers. The epidermis has five different layers. After that, the dermis has two, and we have gone into detail about all of this. And after all of that, we have talked a bit about the accessory structures of the skin, including the nail, the sweat gland, the sebaceous glands. followed by the skeletal biology. We have learned how exactly the bones form from cartilage via the two methods, intramembranous ossification and endochondral ossification. So we have dealt quite a bit of ground this week, and we'll be following it up with problem solving session. Okay, but before going right ahead, we have something in the chat. Okay, Dulab, the thing is that NPTEL, most of the lectures on the NPTEL course will be pre-recorded lectures. So you can go ahead and there will be, when you log in and go to Animal Physiology, then in the course site you will have a particular button called go to course or go to course videos, and when you open that you will see various weeks videos being uploaded in that section, and you can go watch them at your own pace. Just keep an eye out for the assignment questions. The assignment questions are weekly, and they will have a particular time within which you will have to complete them. And you just go through the weeks, you go through the week's videos whenever possible, and then go ahead and solve the assignment questions and submit them. So it is right there on the site. Do you want me to go ahead and show you, or if it is fine, then I will leave it? You can even find them on YouTube actually. If you just search for Animal Physiology and NPTEL, then you can just find the videos directly. What about the assignment of the first week? I think the final date for submitting the first week's assignment is 8th, if I am not wrong. So you have at least one day to finish, so you can so I will just show it to you. So yeah, so this is some so say you are right here, you will be asked to sign in, and then you search for the course Animal Physiology by Professor Mainak Das. If you have enrolled for the course, this button will appear for you. Go to course, and this kind of a page will open, which will have information about the live sessions, and on the left you see a tab having all the week's lectures. So these are the lectures, you can click on them to see the videos, and here at the end of it is the quiz. So you can just go ahead and click on the quiz, and then you will have the first week's assignment. You just solve it and then submit right here. Is that fine? Okay. So quickly going back, yes, we have a lot of people joining. That's nice. So yeah, we have a couple of questions from last week's lecture as well. Very basic questions. So we will quickly go through them. So the first question, so how we will be dealing with this problem solving is that I will show you a question from the assignment. It will have 4 options, and I will ask you to put whatever you think is the correct choice in the chat. Then we will have a discussion on the question, and then we will try to answer it. So the first question is, what is the basic unit of life? The options are cell, tissue, organ, and organ system. You can put whatever you think is the right answer in the chat now. a cell. You can even put an ABCD if you want, because the later ones will have a longer answers. I would like to encourage everyone to try and answer this, because this is very simple to answer. Yeah. My voice is not clear, is it? Can everyone hear me? Hello? I heard a sound. It's okay. I think that was a momentary disconnection. Okay. Okay, that's fine. So, yeah, just a quick. So, I think everyone has answered quite correctly that the basic unit of life is the cell. So, we'll just quickly go through this and then go to the question that was being asked. So, we can divide, as we have learned in the last week's lectures, that physiology can be divided into three major levels of organization. These include the molecular level, the cellular level, uh, and the tissue level. So, starting from the beginning, we would have a start right here, which should be molecules of life that we have covered briefly in the previous lecture, in previous session. These includes the four biomolecules, which are the nucleic acids, proteins, carbohydrates, and lipids. All of these come together to form subcellular organelles, which are supramolecular structures that help the cell to do various jobs, which essentially make up the cell, which is the basic unit of life. Cell, the basic unit of life is cell. Cells aggregate to form tissues, which work similarly. Similar tissues again join up to form an organ that has a function on the organism level. So, for example, here we have the stomach, whose function is for digestion. We have the heart, whose function is for maintaining the circulatory system. We have the brain, which coordinates most of our decisions throughout life. And finally, all of these different organs make up the organ system. Can anyone tell me what is the organ system shown in this particular image? Yes, you are correct. This is the digestive system that we are talking about here, and diff- digestive system shown right here. You are correct, and different. All of these different systems, they come together to form the final organism. So this is essentially the level of organization that we have been talking about time and again. Molecules of life. Let me just check my Wi-Fi if that is working or not. My voice is clear. I think that this is a problem with Almas's connection. I think that's what the problem is here. That is why you're not receiving the voice clearly. Try sitting somewhere else. Maybe that will clear it out. Okay. So finally, from organ system, we arrive at the organism. These are the different level of organization that we have been dealing with. And yes, anyone has a question? Okay. So yeah, for the first question, it was pretty easy. What is the basic unit of life? The basic unit of life is the cell. Okay, so I will quickly go through the question that was posted in the chat. Okay, my voice is coming low. I will deal with that. Just let me go through this question. I will try and deal with that. So there is a question in the chat. Sir, I have joined this course yesterday. Please tell me from where I get the topic of assignment. How to submit my first week's assignment. We just dealt with it. If you go to the main page of the course, then it will be looking something like this. Uh, so if you go to any one of the videos, it will look like this, and in the same tab, okay, if you have go to the quiz section, then it will look something like this, and this is where you submit the quiz. You put in whatever you think is the correct answer and then submit. What I will do is that I will probably move myself and try to be louder. Just wait just a second. Yeah, I just turned it off. I'm moving to the cabin. Bolo. Uh, I need chocolate but don't have the time. Abhi. I will take it later. Yes, so I have moved to the cabin. So now is my voice louder and much more audible? Can anyone tell me? Is this better? Okay, that's nice. Yeah, that's nice. So we'll be going forward. So I think we dealt with the issue that we are facing, and I think someone was asking something. Al Madina, I think that solves your problem as well, right? You just have to go to the course site here, sign in. If you have signed in, you will get option to go to course. After going to course, you have the course outline on the left. From the course outline, if you click on the week that you want to listen to, all the videos are here. Just click on the videos. A video will open and you can go through it, and then after the all the videos are done, you can go to quiz. Week 2 assignment 2 is what we are looking at now, and here you will have all the questions related to week 2. You just put in whatever you think is the correct answer and just go ahead and submit. Okay, what is the last date of first assignment? That all the information is given right here. You just have to go here and see. You can see here the last date for week 2's assignment is tomorrow, so yeah, you should hurry up and finish that. And for week 1, I think week 1 tomorrow is, yeah, for week 1 also it is tomorrow. So yeah, finish as soon as you finish the problem solving session. Maybe you want to go ahead and solve everything and just submit. It will not take more than 10-15 minutes of your time. It's pretty easy. Okay, that being done, yeah, we'll go on to the next question. The plasma membrane is a, there are four options: sulfated monolayer, phospholipid bilayer, rigid structure, and red colored structure. Everyone, you can put in whatever you think is the correct answer. We have a lot of B's, phospholipid bilayer, coming in. Anyone else? Okay, we have flurry of B's coming in. That is phospholipid bilayer. Anyone else that wants to answer this, they can go ahead and answer. Matrudata also says B. Maheshwari, Sibani, Arya, Vishwajit, Saida, Surajit, Akansha, Kamlesh, Ananya, Sanjana, Bisu, Almadina all say it is B. Okay. Anyone else that wants to put in an answer?
Sir, is this question is any assignment question?
So, what we are dealing with here is all the questions that were the previous year's assignment questions. We are not going to deal with questions that are being asked this year. We do not do that. We instead do a previous year's assignment question and try to solve them, because most of the questions are pretty similar-ish, and we actually go through the same concept. So these sessions essentially help you answer this year's questions as well.
Okay, sir.
Okay. So yeah, this is what it looks like, the cell membrane. So we have actually talked about the cell membrane, the constituent of the cell membrane, a bit earlier in the previous lecture. We know that membranes in our cells and organelles are made up of phospholipids. What are phospholipids? Phospholipids are a class of lipids that have essentially two different hands, you can say. One hand is made out of a hydrophilic tail, a hydrophilic head which loves water and is made out of a polar group attached to a phosphate bond, and a hydrophobic tail, which is a long tail of carbon atoms which does not like water. Our the membrane found by the phospholipid, the prime example is the cell membrane. It is a phospholipid bilayer. What I mean by that is that in the cell, in the extracellular space or the intracellular space, both of them have water in them. So what the phospholipids do is that they attach end to end. The hydrophobic parts attach to each other, and the hydrophilic heads aim either extracellularly or intracellularly. So this is what you can see right here. These lines show the hydrophobic tails, which are aimed toward the interior of the cell membrane, and the hydrophilic phosphate groups are the circles you can see, which are aiming either towards the outside of the cell extracellularly or intracellularly. This is what the basic construction of the cell membrane looks like. It is made out of phospholipids, but it is not only made out of phospholipids. There are multiple other things that are attached. First is that there is another type of lipid known as steroid. Steroids are essentially four-membered are molecules made out of four cyclic chains attached, or four cyclic groups attached. Most basic of them include cholesterol, and there is a huge amount of cholesterol present inside the cell membrane. So in the hydrophobic region there is a lot of cholesterol present. Next, there are proteins which are embedded in the cell membrane. These proteins help in majorly in cell signaling, cell transport of stuff from outside to inside or inside to outside, and they can either be halfway embedded or fully embedded in the membrane. What I mean by that is that it is like a tunnel with one end facing outwards and one end facing inwards, or it is like just a boulder on one side. If it is going through and through, it is known as a integral membrane protein. If it is only on one side, it is known as a peripheral membrane protein. Cool? Fine up to here? I guess this is okay. And So we have a majority of the cell membrane made out of a phospholipid bilayer with proteins embedded in it, and on top of proteins there are carbohydrates also, which is the third type of biomolecule that we learned about in the last lecture. And the carbohydrate are actually present on the outer surface or the extracellular surface of the cell membrane, and they essentially help in giving an identity to the cell. So as we have fingerprints to identify ourselves, the type and amount of sugars or carbohydrates present on the extracellular surface of the cell membrane identify the type of the cell, or if a cell is endogenous or a external external foreign cell, that can be decided via the constitution of the carbohydrates present on the cell membrane. So this is how our immune system understands if a cell, such as a bacterial cell, is uh, coming from outside. So this is how they differentiate between different types of cells, our own or something from the outside. Is that okay? We are on the same page, right? Yeah, you can just put in yes, no, whatever you want to in the chat if you have understood or not. I can't just say yes. I guess that is okay for everyone else as well. Okay. Uh, so I will assume that everyone else has also gotten a grasp of what I was talking about. So the plasma membrane is a phospholipid bilayer. Anyone has any questions from this? Okay. Moving on, another very simple question. Which of the following categories does stomach belong to? Is it a cell, a tissue, a organ, or an organ system? Ananya says organ, Sanjana says organ, Akansha says C, Bisu says C, Almadina says organ, Pavitra says organ, Sibani also says C, Surajit also says C, Mutulata also says C. Anyone else? Anyone else wants to try? Okay, so just going back, we just covered this, that we have organelles, cells, tissues, organs, organ systems, and body. Stomach obviously falls into category of organ. Okay, now we go to actually interesting questions that are involved in this week's lecture. So yeah, we are changing gears towards this week's lecture's questions. The first question is, which pigment in our body absorbs UV rays? The options are chlorophyll, melanin, melatonin, and elastin. Akansha, Bisu both say B. Surajit also says melanin. Sanjana also says melanin. Matrudatta also says melanin. Sabani also says melanin. Ananya also says melanin. Anyone else? We have around 40 people in the Google Meet. I think more people can try and answer this. What pigment in our body absorbs UV rays? Chlorophyll, melanin, melatonin, or elastin? Pavitra also says it is Miller. Vrishta also says it is B. Okay. Okay. So let me ask you something a bit different. What do you understand by the term pigment? What do you mean by a pigment? You can unmute yourself and answer this or type in the chat. What do you mean by a pigment? No one has an idea what a pigment is? Sanjana says a colored substance. Avarishta says it is coloration. Bisu says it is color. Ananya says it is a chemical substance that impart color to tissue. Okay. Anyone else wants to try? Okay. So firstly, so the answer to our question, before we answer to our question, the pigment is always the UV absorbing pigment is present in our skin, and the reason for our skin from people to people varying from fair to dark is because of the presence of the pigment. So you are correct, a pigment is a substance which provides color, but the question is how exactly does that work? How exactly does a substance or a chemical provide like have a color? What exactly is that? So what happens is that our pigment molecule, this could be any molecule that can absorb a photon. Photon is energy carrying unit in light, and once a photon is targeted at a pigment molecule, the pigment molecule can absorb that photon, and it reaches something known as a excited state, which you can see right here. So photon from one layer can go to another layer or one orbit to another orbit. Once this happens, the electron can enjoy the excited state for a very short amount of time. Normally it is around nanoseconds, and it then drops back to its normal ground state. Once in the ground state, what happens is that while coming down, it emits another photon. So one photon got it to the higher level, it stayed there for a while and came down. Once coming down, it will emit another photon. This photon will always be a photon of lower frequency than the one that originally hit it. And this second photon is what we perceive as color. So depending on how long the pigment molecule could stay in the excited state, or rather it should be pigment electron stay in the excited state, we have different phenomena such as fluorescence, phosphorescence, or normal color. And depending on which state from the excited state the electron dropped down to, we have photons of different colors, and that is why different objects look like different objects have different colors, like the leaf is green or whatever, the mobile phone is black. This is what the entire idea behind pigments and colors in general. Is that okay? Does anyone have any questions on this? I see a raised hand from Just did you have any question? Hello. Just do you have any questions, or is that clear? Everyone, what do you think? You are clear with what a pigment is, or what exactly color is? Okay. And just quickly going through this, I think you will probably remember it from your class 11, 12 in organic chemistry, that the d orbital containing metals, zinc, copper, iron, all of these have unfinished d orbitals or uncompleted d orbitals, and those are very prone to catching ambient light and jumping up and down, and that is why most of the d orbitals, uh, the d orbital metals are colored. Just brushing up on basic chemistry. Okay, so the answer to our question that we have asked a while back is melanin. Melanin is the pigment that is present in our skin that can absorb UV rays. It and absorption of UV rays helps protect protect the interior cells from harmful UV rays of the sun, which prevent different types of cancers or DNA breaks or essentially cell damage. Melanin is formed from amino acid known as tyrosine. Tyrosine is acted on upon by an enzyme known as tyrosinase and goes through two steps to form melanin. So eumelanin that you see right here is essentially the structure that we mostly deal with. It has three separate ring-like structures right here. And here, one thing that I would like to tell you, that most of the time when we are talking about a UV ray absorber or a light photon absorber, most of the time these are aromatic groups. And here you can see the benzene rings, the aniline rings right here, which can easily absorb UV rays. So most of the time, whenever you think of UV ray absorption or normal visible spectra light ray absorption, most of the time those are aromatic rings that do this. So in the case of melanin, we see the rings right here which can easily absorb the UV rays. And where are they present? Depending on the origin, on your origin, if you are a Caucasian, then most of the melanin that you have is present in the first two layers, the stratum basilae or the stratum germinativum and stratum spinosum. And if you are from close to the equator, then most of the melanin is present in the stratum granulosum. And that is why, depending on how deep the melanin is present, you can actually see it with your eyes, and that actually determines a person's skin color. So the answer to our question, which pigment in our body absorbs UV rays, the answer is
So
melanin.
that's again.
Yeah, Pavitra, are you saying something?
So can you please show that slide of melanin formation again?
Yeah. Is this in the way? I can remove this.
Okay, sir. Thank you.
Okay. Okay, the which pigment in our body absorbs UV rays? The answer is melanin. Anyone else has any questions from this? Okay, I guess not. So we will be moving on. Next question is, metabolism of which of these molecules in our body is directly linked to sunlight? The options are vitamin A, vitamin B, vitamin C, and vitamin D. A, B, C, D. You can put in whatever you think is the correct answer in the chat. Bisu says D, vitamin D. Ananya says vitamin D. Mathuda says vitamin D. Surajit, Akansha, Priti, Sibani, Kamlesh, all are for vitamin D. Al Madina also says vitamin D. Anyone else? Anyone else wants to answer this? Okay, so yeah, all of you are correct. The correct answer is vitamin D. Vitamin D is closely related to sunlight, and that is why most of our friends and parents always ask us to go out in the sunlight because that will help us generate vitamin D. But how exactly does sunlight help in vitamin D formation? So our skin already has pre uh, a premature or a pre-form of vitamin D, which is known as 7-dehydrocholesterol. So as I said, cholesterol is a lipid and makes up most of our like different kinds of steroid hormones and vitamins. And one of the examples is vitamin D. So once you have sun rays hitting your skin, the pre-form of vitamin D, which is 7-hydrocholesterol, gets converted into cholecalciferol, which is vitamin D3. Interestingly, if you have a deficiency in your diet, you are not being exposed to the sun very often, which happens majority in case of lab workers like me, then you will have to take a diet of vitamin D3, which is essentially tablets or capsules with cholecalciferol, vitamin D3 directly. You can take it, and this vitamin D3 is first processed via the liver, which changes cholecalciferol into calciferol, which is one of like an intermediate step, which then goes through the kidneys to form the active form of vitamin D, which is calcitrol. Calcitrol helps in a lot of things, majorly being in calcium absorption, which helps in bone calcification, bone reabsorption, and reduces the excretion of calcium, and also helps in the retention of phosphate. Plus, calciferol can also alternatively help in formation of parathyroid hormone, which is one of the hormones that our thyroid gland produces, which is also important for calcium and phosphate metabolism. So this is all that vitamin D3 does, and it is very important for us. And the major source that a normal person should get it via is the UV sunlight. You step out into the sun, the pre-form of the vitamin D will be converted into the active form normally. So the correct answer is vitamin D. Metabolism of which of these molecules linked in our body is linked to sunlight? It is vitamin D. Does anyone have any questions from this? Okay. Next, we move on to the next question, which is the appendages that connect muscle to bone are called, the options being, ligaments, tendons, cartilage, or nephrons. muscle to bone. So Ananya says these are tendons, Mathrurata says it is tendons, Sanjani Akansha says B, Surajit also says tendons, BC also says tendons, Kamlesh and Al-Madina are also for tendons. So everyone is like, tendons are the appendages that connect muscle to bone. Anyone else wants to chime in? Okay, the question is appendages that connect muscle to bone. Anyone else wants to answer this? The options are A. Ligaments, B. Tendons, C. Cartilage, or D. Nephrons. Okay, so for this we have a short video that we can look at to understand the basic of this. You will just have to let me know if you can hear the sound or not.
विवेक आप गए नहीं
Wait, we have an ad.
विवेक आप गए नहीं
Sir,
that there's no confusion. Ligaments connect bone to do what you need to heal.
Can you hear this? Hello?
You need to understand the basics of those tendons.
Tendon basics.
The tendons are connective tissue made up mostly of collagen, and that's somewhat like ligaments and fascia. Now, let's get this out of the way from the beginning so that there's no confusion. Ligaments connect bone to bone, fascia connect muscle to muscle, and tendons connect muscle to bone. You see, when you move your bones, you contract your muscles. But it's not your muscles that pull on that bone, it's your tendons. All the force goes through the tendons, and that means they have to be extremely strong. But tendons aren't just these strong, static tissues, they also have elastic properties. So if you're jumping or running, they work a bit like a spring, storing some of the energy and releasing.
I can increase the audio. Go back a bit. This is the important segment.
Tendons are connective tissue made up mostly of collagen, and that's somewhat like ligaments and fascia. Now, let's get this out of the way from the beginning so that there's no confusion. Ligaments connect bone to bone, fascia connect muscle to muscle, and tendons connect muscle to bone. You see, when you move your bones, you contract your muscles. But it's not your muscles that pull on that bone, it's your tendons. All the force goes through the tendons, and that means they have to be extremely strong. But tendons aren't just these strong, static tissues, they also have elastic properties. So if you're jumping or running, they work a bit like a spring, storing some of the energy and releasing it on the upward bound. And that's pretty cool. Now let's look at the actual structure of it.
Okay, the audio is not very clear. The audio is not clear, you say. What we can do is that we can essentially share the YouTube link. Yes, I can do that, or I can play it and play it on here on YouTube itself. I think that will be nice. Let me know if you can hear or not. Yeah.
To do what you need to heal, you need to understand the basics of those tendons.
Tendon basics.
The tendons are connective tissue made up mostly of collagen, and that's somewhat like ligaments and fascia. Now, let's get this out of the way from the beginning so that there's no confusion. Ligaments connect.
Hello, am I audible? Okay, now let me know if you can hear it.
To do what you need to heal, you need to understand the basics of those tendons.
Can you hear this?
Tendon basics.
The tendons are connective tissue made up mostly of collagen, and that's somewhat like ligaments and fascia. Now, let's get this out of the way from the beginning so that there's no confusion. Ligaments connect bone to bone, fascia connect muscle to muscle, and tendons connect muscle to bone. You see, when you move your bones, you contract your muscles. But it's not your muscles that pull on that bone, it's your tendons. All the force goes through the tendons, and that means they have to be extremely strong. But tendons aren't just these strong, static tissues, they also have elastic properties. So if you're jumping or running, they work a bit like a spring, storing some of the energy and releasing it on the upward bound. And that's pretty cool. Now let's look at the actual structure of a tendon. Each tendon, like this one, is made up of different bundles. Here's a bundle. There are bundles here, bundles here, bundles here, and finally, here is the collagen fibril. If you have a healthy tendon, it looks a bit like this, with these dense arrays of collagen fibers all parallel to each other. This structure keeps them strong. And opposite of healthy tendons are injured tendons, and tendon injuries are known as tendinopathies. Here's how this works. Tendons are meant to withstand a lot of repetitive loading. But if the load becomes too great, then the tendon gets stressed. They get micro tears. Most of the time, your body can repair these pretty easily, yet if the tendon is repeatedly strained so that the rate of degradation exceeds that of regeneration, that is where the problems start. Now, most of these tendon problems happen near the joints, such as the elbow, shoulder, knee, and ankle. Basically, areas where we put the most strain on. And if you go to a doctor or health professional, you might hear different terms that describe tendon injuries. The most common term is tendinitis, which describes acute tendon injuries that lead to inflammation. Also, one of the most common things that aging athletes get. That inflamed tendon can hurt. Of course, we understand, right, that inflammation is also the body's natural healing response. Your body's working to heal that tendon, and it's totally normal. But we all want to get past that, right? So how do you heal it? Healing. The first step is to make sure you don't injure it more. That's where some degree of rest comes in. Essentially, change up the activities you're doing to put a bit less stress on that tendon. You don't have to totally stop exercising, and just so you know, the initial repair of that tendon will happen in a few days, but to permanently heal it may take more like 3 to 6 months. You just want to make sure you're not overstraining it again at this stage and permanently damaging the tendon. So you have to be a bit careful, and well, a good diagnosis from a doctor is going to help you know what's up. The next step is rehabbing it. Now, some of the best exercises at this stage are going to be eccentric style exercises. That means exercises where you're letting your muscle lengthen, like lowering a biceps curl. That's an eccentric motion. Lots of doctors now recommend moving, stretching, and gently massaging your tendon while it's healing. The goal of this is to make sure that your healing collagen fibers that make up your tendon end up laying down in the right direction, all parallel to each other, resulting in a strong, healthy tendon. And in the end, just remember that getting the right rehab schedule is really important to healing, and to do that, it's important to get a good diagnosis. If you don't think you're adept enough at diagnosing your own injury, then I highly recommend a good orthopedic doctor. I hope
That should be enough. Going back to screen sharing, is that okay for you? Like, he had a very quick guide to what tendons are. Anyone wants to ask any questions from this? Any questions? Oh. Go to the next slide like this. Okay, so the correct answer to, yeah, okay, no questions here. The appendages that connect muscle to bone are known as tendons. Okay, moving on. The next question is very, very similar. The appendages that connect bone to bone are called, the options are ligaments, tendons, cartilages, or nephrons again. Anandya and Sanjana say ligaments, Mattu Datta also says A, Akansha also says A, Ishwarya says tendons, Armadina says it is ligaments, Jasmine says it's ligaments. Anyone else? Anyone else wants to try? What do you think are the appendages that connect bone to bone? Ligaments, tendons, cartilage, or nephrons? Okay, so we had another video on ligaments, but I think I will skip that for now. So essentially, as we had seen previously, that appendages that connect bone to bone are known as ligaments, and the ones that connect bone to muscles are known as tendons. Okay, the next question is, which of the following joints allows for movement in multiple planes? The options are ball and socket joint, hinge joint, pivot joint, or none of the above. Ananya says it is ball and socket joint. Sanjana also says it is ball and socket. Anyone else? Ishwarya also says it is A, ball and socket joint. Akansha also says it is A. Preeti also says it is A. Anyone else wants to try? Jasmine also says it is A. Okay, so here we had a video from Khan Academy. So I would like to hear talk about a bit about this Khan Academy. It is a very excellent channel that you can follow on YouTube, which has a lot about biology essentially, but also different types of other topics as well.
So,
Tell me if you can hear this. Can you hear this?
so ligaments and tendons are types of extra strong and dense connective tissues. Ligaments connect bones to other bones, and tendons connect muscle to bone. And the point where one bone articulates or meets up with another is called a joint. Now, there are different types of joints found throughout the body, and the first type is called synarthroses. which are immovable joints where two bones are fused together. For example, you'll find these types of joints in the skull. When you were a baby, your skull was made up of a bunch of different pieces that grew as your brain grew. And then as an adult, the joints between these bones then fused and became synarthroses. The next type of joint is called an amphiarthrosis. Amphi meaning it's both stiff but also slightly movable. And an example of amphiarthrosis would be your vertebral joints. And then finally we have what are synovial joints, which are also known as diarthroses, of which there are a couple different types. One type, for example, is the ball and socket synovial joint. And you'll find examples of ball and socket joints in your shoulders or in your hips. These are joints that have many degrees of motion. And then another type of synovial joint is the hinge joint. An example of a hinge joint would be, say, your elbow or your knee, where that joint pretty much moves in just one plane, like the hinge of a door. Now, synovial joints are named so because they are lubricated by a fluid called synovial fluid, which is contained within the synovial capsule that surrounds the whole joint. Now, the surfaces of bones that meet up in a joint are lined by a special kind of smooth cartilage called articular cartilage, which is composed of specifically hyaline cartilage. And articular cartilage, like all cartilage, is avascular, meaning it lacks any sort of vasculature or blood vessels. And so it has a hard time getting the nutrients it needs to heal and recover if it were to become damaged by overuse or infection. And as you might be somewhat familiar with, the overuse of joints over time can lead to inflammation, which is called arthritis. From arth meaning joint and itis meaning inflammation. And this can lead to permanent destruction of articular cartilage, which causes the pain and stiffness of arthritis.
The joints of the skeleton.
Okay. So that was about different types of joints. We'll go back to screen sharing. Go back to the presentation. So, as we have just learned, the type of joint that allows for movement in multiple planes is the ball and socket joint. So, someone raised a hand. Tanushri, I think, do you have any questions? Tanushri? Okay. Right. No. Hello, am I audible? Okay, okay. Got afraid for a moment. Okay. Okay, so the joint that allows movement in multiple planes is ball and socket joint. The next question is, human hair is mostly made out of which of the following? The options are carbohydrate, lipid, flax, or protein. So Sanjana, Ishwarya, Ananya, and Muthruratta say it is protein. Akansha, Sibani, Tanushi also say it is a D, protein. Preeti also says it is protein. Happy says it is protein. Almadina also says it is protein. Anyone else wants to chime in? Jasmine also puts in her answer. It is protein. Anyone else? Ishwari says it is keratin protein. You are correct, Ishwari. It is keratin protein. We'll be learning a bit about it. Any questions from this? Anything else? Okay, so yeah, so this is the hair structure of the hair. Hair is essentially a accessory structure to the skin, and it penetrates from all the way from the reticular layer of the dermis all the way to the outside of the skin, and consists in a cross section. If you look at it, consists of three layers. First is the cuticle, or we can go opposite. First is the center of the hair, which is medulla, which are present of live cells. These are filled up of keratin protein. As we move outside, we reach the cortex, where there are dead cells present all around the medulla, and these are keratinized cells. These cells have been amassing so much keratin that they have essentially died, and they form small packaged a small packaged layer, a tightly packed layer of the cortex. And finally, we have the cuticle, which is the outermost layer, which is the exposed surface of the hair, and this is shed continuously and replaced by new medullary stem cells that keep on propagating and dividing and elongating the hair. And here on this side we have a schematic of the hair fiber. As we had just seen about the tendon, the hair fiber also is iterative. It is essentially iterative in the sense that it has layer upon layer upon layer of coiled fibers inside it. So first, if we look at the cross section of the hair here, if you look deeply, you will see it is made out of tightly packed cortical cells. These tightly packed cortical cells essentially have tightly packed microfibrils inside them, which are made out of complex keratin, like complex keratin. This microfibril, if you look even deeper, it is made out of intermediate fibril or intermediate filament, which is essentially made out of complex keratin. So essentially, this is a repetitive formation of compacted and even more compacted keratin that is present inside the cells. So just a takeaway point: medulla is made out of live cells, cortex dead cells very tightly packed together, and the cuticle only has keratin on the outside, like it is the exposed layer, and it is just made out of keratin. Any questions from this? Okay. So human hair is mostly made out of the keratin protein. Just a quick point: keratin. So does anyone know keratin has essentially just one type of secondary structure? If you know this, if anyone has come across, keratin has essentially one type of secondary structure. Does anyone know what that is? Okay. So keratin is made out of mostly alpha helix. The entire protein is made out of mostly alpha helices. So if you have done a course on biochemistry, then you will know that protein structure has different levels. As we have discussed in the last class, the most basic unit of a protein is known as an amino acid. There are 20 different amino acids that can be linked together via something known as a peptide bond. So essentially they can form a long chain of peptide bond, one after the other, one after the other, and these chains can be huge. That is known as the primary structure, where we have information about what amino acid follows another. So it could be a combination of any combination of the 20 known amino acids that code for a protein. That is a primary structure. But inside the cell, the protein undergoes something known as folding. So just the primary structure of the protein is not essential for its function. How it folds upon itself to form a three-dimensional structure is important. in order to form a three-dimensional structure or a tertiary structure, first the protein goes through a secondary structure, which is a localized folding, and this can be of different types. It can be alpha helices, beta sheets, alpha helices or beta sheets, and these can form other higher secondary structures such as beta barrel, beta helix, all of these things, which form the tertiary structure. Tertiary structure onwards, the protein starts to gain function, but actual function of the protein is when different parts of the proteins interact with each other in a quaternary structure. So 4 levels of organizations are present for the protein. And for keratin, the entire protein is made out of only one kind of secondary structure, which is the alpha helix. Is that fine? Okay. So if this question is okay, we'll going on to the next question, which is which of these is not a muscle type? The options are skeletal muscle, cardiac muscle, smooth muscle, or rough muscle. So here's the answers pouring in. Akansha says it is D, rough muscle. Sanjana says it is rough muscle. Happy, Ananya, Ishwarya, and Preeti all say it is D, rough muscle. Anyone else wants to try and answer this? Jasmine also says it is D, rough muscle. Almadina also says it is a D, rough muscle. Okay. So yeah, so you are all absolutely correct. There is nothing known as a rough muscle. There are 3 types of muscles present in our body: skeletal muscles, cardiac muscles, or smooth muscles. Here is a brief comparison between all the 3. So firstly, location: skeletal muscles are present attached to bones or skin of the skin of the face. And so this just goes along with its name. It is attached to the skeleton. Cardiac muscle also goes along with its name, is completely related to the heart, so it is present in the walls of the heart. And finally, smooth muscles, which are present inside hollow organs, especially in our digestive tract, such as the stomach. So it is present inside in the inner lining of this. What exactly gave these names, skeletal, cardiac, or rough, is actually the histological anatomy of the muscles under the microscope. So if you look at the skeletal muscles, there is a single very long cylindrical, striated, and multinucleate. A lot of words, but essentially these skeletal muscle cells are essentially one particular cell is very long and has multiple nucleus in it. The reason for it is that when this muscle was initially forming, it was made out of different muscle cells, but as development proceeded, all of them fused together to form a long cell which has multiple nucleuses because they originally were very different cells. So skeletal muscles are single, very long, cylindrical, striated. The striated pattern is due to a particular orientation of a protein band known as the Z band. We will learn more of this in the next lecture, or in the next week's lectures. What are these striated bands? And yeah, so this essentially makes up the skeletal muscles. Second type of muscles are known as cardiac muscles. These are branching chain of cells, so these are not fused together, striated and uninucleated. So these guys remain separate, they did not fuse together, so they are not very long and have various branches, but still have one nuclei each and are striated still. So they still have the Z band pattern. Finally, we have smooth muscles, which are single, fusiform, no striation, and uninucleate. So these guys also did not fuse together. They are also different, and there is no striation, that is, there is no presence of a Z band. So we have a question here: does having multinuclei have any significance, like in providing more energy or whatever? you are part correct. So essentially the nucleus is because there were multiple cells which were close by together and needed to fuse together and form one single cell. The reason for there is a point that you make that is correct. The muscle cells require a lot of energy. How do they exactly make up for that energy? Is they have a huge amount of endoplasmic reticulum and mitochondria. The endoplasmic reticulum, which in this case is specially named sarcoplasmic reticulum, is essentially fused together between all the cells and he forms a huge complex. And this sarcoplasmic reticulum along with the mitochondria have a special type of signaling that goes on via calcium ions, which provides instantaneous transmission to skeletal muscles, because obviously they have to act fill very fast and provide a lot of energy to the cells. So they provide energy, but not the multinucleus thing is not the reason for it. Is that fine? Okay. And finally, we have regulation of contraction. Skeletal muscles are voluntary. That is, we can control when they are being contracted. These are controlled via the nervous system. The cardiac is involuntary. The heart has its own pacemaker. So pacemaker is essentially something that sets the rhythm for the heart. Does someone know what the pacemaker of the heart is known as? Anyone has any idea? Absolutely correct, Akanksha. It is SAN. It is the, can anyone tell me the full form? Cyanoatrial node. Yes, cyanoatrial node is the full form of SAN. It is present about at the top-ish part of the heart where the atrials fuse, and this set of cells essentially dictate the rhythm at which the heart beats. That is why a pretty interesting and pretty unique way of controlling the beating of the heart, but we will get into it when we get into the later lectures. It is a very, very interesting thing that we will get into. So the cyanoatrial node, as you were saying, is the correct answer. It is the heart's pacemaker. And finally, smooth muscles. Smooth muscles present in the digestive tract or anywhere else are involuntary. These guys are to do their own thing and do it via the nervous system or via hormones or other chemicals secreted by their surroundings. So essentially these are the three different types of muscles: skeletal, cardiac and smooth. So which of the following is not a muscle type? The correct answer is rough muscle. Okay, so this is all we had for today about the problem solving, but I do have another video on the skin. So since we have time, I will just quickly play this, and we will just be will be a repeat of what the lectures were, but you have a better coagulated idea of what exactly it is, and you can better for revision as well. Just go ahead and type this in.
They say that beauty is only skin deep. But
is this audible? Okay. Let's remove this.
But did you know that the outermost layer of your skin is actually composed of entirely dead cells? I have no idea what's beautiful about dead tissue, but in this video, we'll seek to figure that out. We'll talk about the different layers of skin. As you can see here, there are many layers to the skin, and there are 3 different sets of layers that we can talk about. There's the epidermis, which is the first 5 layers, the dermis, which is the next 2 layers, and then the last part, the subcutaneous tissue, or the hypodermis. In this video, we'll start by talking about the most superficial part of your skin, and that is the epidermis. And I'm sure your friends have told you before that your epidermis is showing. The epidermis is the topmost layer of skin, and itself is comprised of 5 layers, or as we call them, strata. So 5 layers or strata, and each strata or stratum has its own important characteristics. So we'll start from the bottom and talk about the deepest or bottommost layer of the epidermis, and that's known as the stratum basale, or the basal layer. The stratum basale sits right above the dermis and is the place where we first generate what are called keratinocytes. And so I'm drawing a whole bunch of keratinocytes right here. These are keratinocytes. And in fact, the stratum basale has multiple layers of keratinocytes. And as you might recall, cyte just means cell. So what does keratino mean? Well, it comes from a protein or an intermediate filament that's called cytokeratin. Cytokeratin, which is something we first start making in the stratum basale. Sometimes it's just called keratin. And it helps give our skin its tough outer layer when we get to the top, as we'll talk about in a minute. And in other animals, keratin actually is the main ingredient for things like horns, or even hooves. So it's definitely the tough stuff that help protect our skin. And another very important thing to know about the stratum basale is this is where we have very rapid cell division. Rapid cell division because this is the bottom layer of our epidermis, so cells are made here and slowly they move upwards. Another thing that's important about the stratum basale is this is where we get our skin color from. So there are components of the stratum basale that determine what our skin color is going to be. And the cells that confer skin color for us sit here as well. And these guys, you may have heard about them before, are called melanocytes. So melanocytes, and as you know, cytes again just means cell, but melano just indicates that there's a special pigment that's made in these cells that helps determine our skin color. And that pigment, as you may have guessed, is called melanin. And the interesting thing about these cells or this pigment is that it's not the number of melanocytes that determine how dark your skin is. In fact, people with dark skin color and light skin color have the same number of melanocytes. It's actually melanin, the amount of melanin that you have, that determines how dark your skin color is, meaning that darker skinned people have more melanin. Alright, so as we move up to our next layer over here, we see that our keratinocytes make it up here too, and we might start to notice some of this cytokeratin that's being produced here a little bit. And one of the unique features about this next layer is that we have this little connection that's starting to be generated in between our keratinocytes. This guy is called a desmosome. And this is involved in what gives this layer its name. So this layer that sits above the stratum basale is called the stratum spinosum. Which, as you might guess from the fact that we call this our basal layer, this stratum is our spiny layer. And the reason why we call it that is because of the desmosome. When we look at this layer of skin, sometimes underneath some microscopes where we have lost moisture or the water that is in our cells, we'll have these shrunken cells that actually look like stars or kind of like spiny cells, very pointy shapes. And the reason why they're pointy is because they're still connected by these desmosomes, but they've lost the water. So that's why sometimes these layers looked shriveled. So shriveled because they've lost water, as well as spiny. And so that's how we remember the name of the stratum spinosum. Another type of cell that sits in this layer is an immune cell that is looking for foreign bodies or pathogens or things that can be destructive to our body like bacteria and fungi and eat them. These are called Langerhans cells. Langerhans cells. And as you may have guessed, these are part of our immune system. So they hang out here but can definitely travel to other layers, and we talk about them more in our dedicated immune system tutorial. Alright, and so it's onward and upward to our next layer that's here. This more superficial layer is called the stratum granulosum. The stratum granulosum, which just means the granular layer. And the reason why we call it this is because the keratinocytes that sit here, now in addition to the desmosomes that we talked about from the spiny layer, have a tremendous amount of granules that are being produced here. And these granules are very apparent when we look at them under a microscope. These granules are called keratohyalin granules. So keratin hyalin granules. And I know what you might be thinking. So does this mean we're making a whole bunch more keratin in this layer? Well, we are, but these granules don't actually house keratin or the cytokeratin we talked about. They hold a whole bunch of other proteins that help handle our keratin. Just know that these granules make proteins that are best remembered as your keratin handling proteins. So your keratohyalin granules make your keratin handling proteins because they move your cytokeratin around in the cell. The other thing the stratum granulosum does is that it releases these things that are called lamellar bodies. And these lamellar bodies contain a whole bunch of lipids that then come together and form a strong lipid layer that sits at the top of our skin. This lipid layer is impermeable. It gives the skin its watertight capabilities that prevent foreign pathogens from making their way down deeper into our skin and eventually into our bloodstream. And so from there we go to our next layer up to the stratum lucidum. The stratum lucidum. And sure enough, this is our lucid layer, or our clear. I'll write that here. This is our clear layer. And the way I think about it was that in the stratum granulosum, our keratinocytes worked so hard to make these keratohyalin granules and release these lamellar bodies that they actually died by the time they make it up to the stratum lucidum. So these cells that we have here, these keratinocytes, are actually dead. These are zombie keratinocytes. And they look that way on microscope because they've lost their nuclei or other organelles that usually gave them the color we saw at our earlier, more alive layers. And so as a result, these layers will not have these granules or nuclei or organelles that we see in the previous layers. Instead, they're going to be far clearer or more see-through than what we saw earlier. And that's an important note to make off to the side. The stratum granulosum has lost their nuclei and organelles, which are usually supposed to give our cells color when we look at them under a microscope. And this trend continues when we move on to our topmost layer here. This is the stratum corneum. The stratum corneum, our topmost layer of the epidermis of our skin entirely. And the way I think about this is that this is the coroner's layer, the coroner. And as you know, the coroner's office is where dead bodies go for autopsies, and so this is also a layer of dead skin. And one of the key characteristics of the stratum corneum is that we'll have stacked layers of our keratinocytes, our dead keratinocytes, and we can have up to 15 to 20 stacked layers of these flat, simple squamous epithelial cells. So I'll write that off over here. We can have about 15 to 20 layers of our keratinocytes. Again, they're still dead because they died in the stratum lucidum. And because this is our topmost layer, we'll see that these cells will randomly as well as continuously slough off. So they will randomly and continuously slough off or leave, they'll fall off. And that makes way for newer cells underneath or newer keratinocytes to make their way up to the top. And another interesting thing about this layer is that reptiles, I'll write this in like a reptile green, so reptiles will molt this layer or shed this layer off in one piece. And so that's why you'll see like snake skin that comes off as a single intact covering to the snake, so they molt off the entire stratum corneum, which is very interesting and also very gross. Alright, so those are all your layers of your epidermis, all 5 of them, the top 2 being dead, and these bottom 3 or deeper 3 layers of the epidermis being alive. And as we move deeper down and start to talk about the dermis, those cells are also alive and have vessels in them to nourish them. We don't have vessels anywhere in the epidermis because our nutrients and our oxygen come from below and make their way upwards.
Okay, so that was very interesting take on presentation of the various layers of epidermis. I hope you had gained an interest from that and learned something interesting. At least it will help you to coagulate. Yeah, happy, yes, it was really interesting, nice. So yeah, it will help you coagulate all the information that you had received throughout the lectures together, and help you prepare, and check out that channel Khan Academy, that has a lot of interesting information about various subjects and might help you prepare for future exams, anything. Okay, so with that I would stop this, close the screen sharing. I think that is all I have for today. The recording of the meeting will be uploaded uh, to my YouTube channel. You can find that on the same website as the NPTEL course, and you can just go ahead and look at it, and the PowerPoint presentation will also be available there, and I will be seeing you next week. Thank you everyone for joining. See you next week.